Children, welcome to the online classes of biology. So, children, we have studied what happens during double fertilization and why triple fusion, the term is there. All that we have seen. After triple fusion, okay, the product of triple fusion is primary endosperm cell, okay, and the product of syngamy, okay, syngamy means fusion of male and female gametes is what? Zygote, isn't it? And primary endosperm nucleus will develop into the endosperm and zygote develops into embryo. Understood? So this is happening. Okay. Now all this we can study under a heading, that is post fertilization events. So what is post fertilization events? That is after double fertilization. Okay, events of endosperm and embryo development, maturation of ovules into seeds and ovary into fruits, all together we can call it as what? Post fertilization events. As I told, the endosperm development is proceeding from where? From the means it is embryo is developing from the primary endosperm nucleus, isn't it? And the primary endosperm nucleus will divide and form the endosperm, and endosperm formation will be taking place first before the embryo development. Why? Because primary endosperm cell divides repeatedly and form a triploid endosperm tissue, and that will have what reserve food materials so that it can give nourishment to the developing embryo. So this can be asked, the among this, okay, after double fertilization, which cell will get developed first? PEN, isn't it? Primary endosperm nucleus will undergo continuous division, and that will result in the formation of endosperm. So this will take place first because this endosperm gives, isn't it, the nourishment to the developing embryo. Okay, and the cells of the endosperm, what is the speciality? They will be filled with reserved food materials. Okay, and that can be used by the developing embryo. Okay, and when you see the endosperm development, okay, you can see that in most of the cases the primary endosperm nucleus will undergo divisions continuously and form a number of nuclei. Okay, so this will give rise to free nuclei. Free nuclei means plural of nucleus only. Okay, number of nucleus will be formed. Okay, so this stage of endosperm development is called free nuclear endosperm. Okay, so there are different kinds. Okay, in some of them you can see along with the nuclear division, cell wall formation also will be there, and so it will be forming cells. But in most of the cases, how it is, first the nucleus will divide continuously. Okay, the nucleus of the PEN, primary endosperm nucleus, divides continuously and form free nuclei. Understood? And afterwards only cellularization, that is how cellularization happens, by the formation of cell wall. Okay, cell wall formation will be delayed. First what will happen? All the nucleus of the primary endosperm nucleus will divide, okay, and form a number of nuclei, and afterwards what will happen? Cell wall formation takes place, and that results in the cellularization. Okay, so have you heard about any, or do you know any endosperm? Familiar endosperm as the coconut water from the tender coconut is actually the free nuclear endosperm. Okay, actually it contains what? The free nuclei only, and the white kernel that we used to eat, that is actually cellular endosperm. So depends upon the different methods of formation. Okay, endosperm formation is of different types that you will learn in higher classes. Okay, so the common one is what? Free nuclear type. Okay, endosperm. So here what happens? The nucleus of the primary endosperm nucleus divides continuously and form what? Free nuclei. Okay, and it will be afterwards only cell wall formation takes place, and they will get the cellularization. Okay, they will be forming the cells. Okay, so the tender coconut, in the tender coconut, the coconut water is actually free nuclear endosperm. Understood? And that the other parts, the white part surrounding this is the cellular endosperm. Okay, and this endosperm, I told it is, what is the main function of endosperm? To give nourishment to the developing embryo. Understood? So before seed maturation, understood? So before the seed maturation happens, embryo development is taking place inside the seed. So in that condition, who gives nourishment? Endosperm gives the nourishment. So in some of the plants, okay, they'll be consumed totally. Okay, totally the endosperm will be consumed by the time the embryo get matured or seed get matured. And in some of them, okay, it may persist. Okay, the endosperm will be persisting in the mature seeds also. Example is what? Castor seeds and coconut seeds. Okay, so in most of the monocots, they are endospermic. The seeds, if they are having endosperm, we call it as what? Endospermic seeds or albuminous seeds. Okay, so most of the monocots are endospermous seeds, and most of the dicots are non-endospermic, okay, or non-albuminous seeds. But castor seed is an exception. Castor is actually a dicot, but it is having endosperm persisting in that. Understood? So like this, if the endosperm is persisting, okay, after the maturation of the seeds also. So usually what happen in dicots and all, when the embryo develops, they utilize the endosperm, okay, and the embryo get matured. Understood? So in some of them, some part of the endosperm will be left behind, and these seeds are known as endospermic seeds. So this much is clear. So about the endosperm development, you understood now. So next is what? Embryo. Embryo is developed from what? The zygote. Zygote is formed where? In the micropylar region, isn't it? Micropylar region only what is there? Egg apparatus and egg. So egg will get fused with the one of the male gamete and produce what? Zygote. Understood? And zygote will develop into what? The embryo. So usually I told already, embryo development will be taking place after the the endosperm development starts, okay, or most zygotes divides only after some amount of endosperm is already formed, okay, so that it can get nourishment from the endosperm. And you can see that the dicot embryo and monocot embryo are entirely different, but the early stages of their development is similar. Okay, so you can see that the zygote will give rise to the proembryo. Then you can see it will get divided into a globular type. You can see here, isn't it? Globular type of embryo, then a heart-shaped embryo, and then you can see what? A mature embryo. So this is the the diagram of a mature embryo. This diagram also you have to learn. Okay, so it will have what? You can see a radicle, then two cotyledons will be there, isn't it? And then this part is the plumule. So as you know, the radicle will give rise to the root system, isn't it? Then the plumule will give rise to the shoot system, and cotyledons, they are actually storing the food. So now you understood, for the development of the zygote into this embryo stage, the mature embryo stage, then they needs energy, isn't it? They need food, reserved food. So that who is providing that? The endosperm. Understood? So till this maturity of embryo takes place, understood, in most of the cases the endosperm will be fully utilized, and in some seeds I told it will be left out. Understood? So this is what this diagram is, actually the structure of a mature embryo. So this you have to learn. Okay, so this stages are showing means these diagrams are showing the different stages by which a mature embryo is formed from a single celled zygote, isn't it? Zygote is a single celled one, isn't it? It's a single cell, and from that only the different stages are formed. First, what will happen? It will form a globular type of embryo, then it will form a heart shaped one, and then it will be converting to a mature embryo. Understood? Mature embryo, and it will have what? Radicle, plumule and cotyledons. Okay. So about the formation of embryo, you understood? So a typical dicotyledonous embryo, isn't it? There are two different types of embryos. What is that? Dicotyledonous and monocotyledonous. Okay. So in the dicotyledonous embryo, what all are the parts? That is, I told plumule and radicle. Okay, that comes in, or we can call together them as the embryonal axis and the two cotyledons. Okay, to the embryonal axis only these cotyledons are attached. What is the function of cotyledons? They help in giving nourishment. Okay, to whom? To the seeds. The embryo will germinate, isn't it? Once you see the sow the seeds, what will happen? The embryo will start growing, and till it will get a root system, proper root system and shoot system to do photosynthesis and also to absorb materials from the soil. It needs food, isn't it? So that is provided by the cotyledons. Understood? So this cotyledons are attached to the embryonal axis, and the portion of the embryonal axis above the cotyledons, above the level of the cotyledon is known as epicotyle. And okay, means between or the portion between the plumule. Plumule is what? The tip, isn't it? Which give rise to the shoot system. And just below that, two or the space or the place between the plumule and the portion of the embryonal axis to which the cotyledons are attached, that is known as epicotyle. Okay, and the cylindrical portion just below the level of cotyledons, okay, is known as hypocotyle, and at its tip, what is their? Radicle or root tip. Okay, and this tip will be covered with a root cap. Okay, root cap will also be there. So this is the, see here the diagram is given in this way, and you can draw the diagram of this if the diagram is asked for explaining, or draw the diagram of a mature dicotyledonous embryo. If it is asked, you can draw this diagram. Okay, so in that you can show plumule. Okay, then cotyledons are attached here, and this part, okay, this part where what the place between or below the plumule and the place where the cotyledons are attached to the embryonal axis is known as epicotyle, and hypocotyle is what? Where the cotyledons are attached to the embryonal axis and between the radicle. This is the radicle, and in between this place is known as the hypocotyle. Understood? Hypocotyle, and you can see radicle. Radicle will be from radicle only. What will be formed? The root system, and there will be a root cap which protects the radicle and the young roots which are growing through the soil. Understood? If you see the structure of a monocotyledonous seed, okay, monocotyledon means what? It will have only one cotyledon, isn't it? One cotyledon only it will be having, and that one cotyledon is known as scutellum. What is the name? Scutellum. Okay, the cotyledon is known as scutellum, and it is seen attached to one side of the embryonal axis. So this is the embryonal axis, and to its one end only it is attached. Okay, and you can see the embryonal axis is having the shoot apex or the plumule, and it is covered by a coleoptile. Okay, can you see here? This is shoot apex, and this is actually the coleoptile. Okay, and the portion just like that, the dicots, the portion above the region of attachment of the cotyledon to the embryonal axis is known as epicotyle. Okay, and just the portion which is just below the attachment of the cotyledon to the embryonal axis is known as the hypocotyle, and it will have it will end in a radicle, and also there will be a root cap, and this will be protected by a coleorhiza. So this is coleoptile. Okay, this part is known as the coleoptile, and the single cotyledon that is known as the scutellum. Then just what below, okay, that coleoptile covers the shoot apex or the plumule, okay, which give rise to the shoot system. Okay, then you can see what the radicle which is found below the hypocotyle or at hypocotyle enters, okay, or it ends in what radicle, and it is provided by a root cap, and this all structures are protected by coleorhiza. Okay, in angiosperms, okay, after sexual reproduction, the product or the thing that formed is the seed, isn't it? So what is actually a seed? Seed is a matured ovule, isn't it? It's a fertilized ovule. And where you can see is seeds in the fruits, in the angiosperms inside the fruits, isn't it? That's what we call it as angiosperms. Gymnosperms, we call it as what? Gymnosperm, because their seeds are naked, but here the seeds are inside the fruits. So when you see a seed, they consist of seed coats, okay, cotyledons, and an embryo axis, isn't it? Then cotyledons are what? They are generally thick, and they store the food materials, isn't it? And they give nourishment to the developing embryo. Means after actually seed, when we are sowing the seed, okay, till the plant or the embryo get what grow into different parts of the plant, till that, okay, who gives nourishment? The cotyledons gives the nourishment. Okay, so if the seeds are having, I told, the residual endosperm, isn't it? Then it is known as albuminous seeds. And if they are not having any residual endosperms, then we call it as what? Non-albuminous seeds. In some seeds, okay, nucellus also persist, and the residual persistent nucellus is known as perisperm. So do you remember what is nucellus? Where it is found? Inside the ovule, isn't it? When the ovule is young, I told it is filled with a tissue called nucellus. Okay, when embryo sac develops, nucellus will be utilized. Understood? And in some seeds, like what? Pepper seeds, okay, then what? Beet, etc., little bit nucellus will be persistent. So that persistent residual nucellus is known as what? Perisperm. Okay, so seeds can be perispermous seeds, means what? They are having remnant nucellus in them. If they are said to be albuminous or endospermous, what is the meaning? They are having the endosperm left out in them. Understood? So integuments, I told, ovules are becoming the seeds. So integuments of seeds, they get converted into seed cods. Okay, and you can see the micropyle. Micropyle will be just remaining as a small pore in the seed coat. In pea seed and all, if you see, you can see. Okay, so what is the function of this small pore? That is, they help in the entry of water, oxygen, etc. during the seed germination. Okay, when the seed matures, what will happen? Its water content will get reduced and they'll become dry, isn't it? And you can see that the metabolic activity of the embryo will also be slowing down, and they may enter into a state known as dormancy. Okay, so if favorable conditions comes back, they will start germinate again. Okay, so what is dormancy? It is a stage in the seeds, okay, or the seeds undergo a stage called dormancy, where what happened, the metabolic activities of the embryo will be slowed down. Okay, and this is necessary in many cases of plants, because during this stage only the seeds will get, the embryo will get matured. Okay, what we have studied today is about the seed. What is a seed? Seed is a matured ovule. Okay, in ovule, I told, the integuments are becoming the seed cods. When the seeds are forming from the ovule, the integuments of the ovule is becoming the seed coat. Okay, and the micropylar region will be remaining in the seed as a small pore, so that it can help in the entry of water and oxygen, etc. during germination. Then if the seeds are having endosperm left out in them, they are known as endospermous seeds or albuminous seeds. Okay, and this usually I told all the dicots are what X albuminous seeds. They won't what remain in them, there won't be any endosperm left out. But in case of which one? In case of monocots, they are albuminous seeds. Dicots are non-albuminous. Okay, or X-albuminous or non-endospermous seeds. All these terms are very clearly you have to understand. Okay, then if the seed is having what remnant of nucellus, then it is known as that nucellus is known as means left out nucellus is known as perisperm. Okay, and that kind of seeds can be called as the perispermous seeds. So about seed you understood now. Now we will have to study about the fruits. Okay, so this much will be very clear to you, I hope. Thank you very much.